Scientists sequenced the entire human genome in just under 4 hours, breaking a new record that could change how quickly doctors diagnose genetic illnesses in critically ill newborns. The achievement sets a Guinness World Record for the fastest human genome sequencing ever completed. To put it in perspective, the original Human Genome Project took over 10 years and nearly $3 billion to finish just one full sequence, completed in 2003. Now, in the time it takes to watch a long movie, researchers can read all 6 billion letters of a person's genetic code and get actionable results. This matters most in neonatal intensive care units, where babies with rare genetic conditions often need urgent answers. Before this, even the most rapid methods still took days to go from DNA sample to diagnosis. The new system cuts that window dramatically. In their pilot, the team tested 15 samples, including newborns and previous rare disease cases. They used a prototype sequencing technology and showed it could reliably deliver results in hours, not days. In one case, they completed everything in less than 4 hours, from extracting DNA to analyzing genetic variants. That speed could prevent unnecessary surgeries, guide medication choices, or even save lives when time is tight. The new workflow could allow hospitals to collect a sample in the morning and have a diagnosis ready before the end of the day, a shift that would be game-changing for families waiting on answers, and a crucial step toward truly personalized medicine, where treatment decisions can be tailored to a baby's exact genetic makeup within hours.
Humans are constantly shedding tiny pieces of DNA from sweat, spit, blood, and skin. But a new study showed that human DNA can be easily collected from air, sand, and water, raising a host of ethical questions about privacy and consent. Initially, the scientists set out to collect environmental DNA or eDNA from sea turtle tracks to study viral tumors affecting these animals. While they anticipated finding genetic material from various species, including humans, they were still determining the informative value of the human DNA they would obtain. The team scooped up genetic fragments from oceans, rivers, and sand in Florida. They also traveled to a remote island rarely visited by humans and gathered samples from the sand there. They found high-quality human DNA for analysis and sequencing in all locations except the remote island. They also retrieved DNA from footprints made in the sand by 4 volunteers. With permission, scientists were able to sequence part of the participants' genomes. Next, the researchers took air samples from a 280-square-foot room in an animal clinic where 6 people worked as they went about their daily routines. The team recovered DNA that matched the staff volunteers, animal patients, and common animal viruses. From the eDNA collected, researchers identified genetic variants associated with European and Latino populations, and variations associated with various disorders and diseases, such as autism, diabetes, eye diseases, cancer, and cardiac diseases. The implications of this breakthrough are immense. The eDNA collected could potentially aid in locating missing persons, assist in forensic investigations, uncover archaeological gems, and even monitor an individual's health. However, the ability to capture human DNA from the environment could have a range of unintended consequences, both inadvertent and malicious. These include privacy breaches, location tracking, data harvesting, and genetic surveillance of individuals or groups. It could lead to ethical hurdles for the approval of wildlife studies.
A new DNA-based computer that can run over 100 billion different simple programs has been developed by scientists. By mixing and matching DNA strands, the researchers created circuits that function like those in conventional computers, but with DNA molecules acting as wires. These circuits, called DNA-based programmable gate arrays, or DPGA, can solve complex mathematical problems, such as square roots and quadratic equations, by chemically interacting with specific molecules. Inputs are added through molecules that shape chemical reactions, similar to how electrons move through wires, and outputs are measured by fluorescent markers. The team demonstrated the potential for real-world applications by designing a DPGA that could classify RNA molecules associated with renal cancer, highlighting the technology's promise for diagnosing diseases from within biological systems. This innovation could pave the way for highly efficient diagnostic devices capable of processing vast amounts of data directly in cells or body fluids. DNA-based technology can perform a full range of data storage and computing functions, including storing, retrieving, computing, erasing, and rewriting data. New methods can now enable DNA to store large amounts of data in small spaces, such as holding the equivalent of 1,000 laptops worth of data in a space the size of a pencil eraser. The work lays the foundation for future molecular computing, offering a cheap, practical, and sustainable alternative to conventional computing technologies.